Hey everyone, it's Ryan, and welcome to another edition of Faculty in 15. It's the podcast where we give one University of Guelph faculty 15 minutes, give or take, to answer 15 questions about their work, their expertise, and a little bit about themselves. Today I'm happy to be joined by Dr. Melanie Wills. Dr. Wills is an adjunct professor in the Department of Molecular and Cellular Biology within the College of Biological Science at the University of Guelph, as well as the director of the Magnotta Research Lab, where she leads research on the diagnosis, prognosis, and treatment of Lyme disease and the development of rapid, reliable, and accessible diagnostic testing. Her work has been recognized with numerous awards, including the Governor General's Academic Medal. Most recently, her team created a groundbreaking biosensor able to detect the tiniest traces of Lyme disease biomarker in a blood sample, an incredible breakthrough with the potential to revolutionize Lyme disease diagnostics. Welcome to the podcast, Dr. Wills. How are you today?
Thank you so much for having me. I'm great.
All right. You got 15 minutes?
Let's do this.
So, first question, what is the biggest misconception people have about Lyme disease?
I would start right at the beginning and say that people sometimes come into this with the misconception that Lyme disease is easy to detect and easy to treat. So we have a bacterium, we've got an antibiotic, problem solved, right? And sometimes that actually is the case. We can see a very clear phenotype or presentation of the disease. And then if that's diagnosed and we come in early with an intervention, people can completely resolve. But that's not the only possible outcome here. And so there's numerous trajectories that patients could end up on, which leads to the second misconception that Lyme disease is always resolved, that there is no long-standing complications that come with Lyme disease. That's been an extremely controversial and polarizing topic. But essentially, we do know now, there's a lot of scientific evidence behind this saying that yes, Lyme disease can cause lingering symptoms, lingering complications. We don't know the mechanism. That is still hotly contested. But unfortunately, not everyone who gets Lyme disease can resolve it easily.
And so why is Lyme disease so difficult to diagnose?
It starts with the fact that when you first get Lyme disease, so it's a disease that is transmitted by a tick. It is bacterial, so it's an infection. When you first get that, you can have a really obvious presentation, which we would call the bullseye rash. So you're literally marked with a bullseye. And if that happens and it's correctly diagnosed, then as I mentioned, you can go on and have sort of a textbook intervention. But for many people, that's actually not the case. So only about 9% of people would get that very classic bullseye presentation. For a lot of other people, it might look really indistinct, which makes it hard to diagnose. It might not show up at all. So there's no rash. And then the early symptoms would be flu-like. So they're generic. They're not specifically indicative of Lyme disease. And so people can't unequivocally pin it on Lyme. And so you end up wondering what has gone on. People often don't notice a tick bite either. So then we get into the question of using laboratory diagnostics. And classically, this has been an indirect approach. So the test that's on the market today actually looks at the patient's immune response. So whether or not the body has seen the pathogen, it's not looking for the pathogen. And this leads to another set of limitations in that approach and another set of opportunities to miss the diagnosis. So the ideal case is to develop a direct test that's going to read or see the pathogen directly. And this is hugely problematic because when people have Lyme disease, the amount of pathogen in the system is usually very, very small. It's below a threshold for detection. So trying to read this needle in a haystack is really problematic.
And so what first drew you to the University of Guelph and what keeps you here?
What initially drew me, well, I came from my undergrad and I loved the fact that the depth and the breadth of the courses that were offered in my field. So I was in MBG, molecular biology and genetics, cell biology, microbiology. Guelph was offering a set of courses at a level that rivaled much bigger schools, but in an environment that was small and community oriented and intimate. And I thought that was fantastic. So that's what got me to Guelph initially. And Guelph has also been a fantastic place to launch a Lyme disease research lab and to also to grow it and evolve it, because Guelph has placed a priority emphasis on One Health, which is this idea of human health, environmental health, animal health, all intersecting in these complex problems. And Lyme disease is the quintessential One Health issue. So having a Lyme disease research lab anchored in a school that prioritizes One Health is a natural fit. And we also have world-class instrumentation that we plug into in the College of Biological Sciences Advanced Analysis Center that really helps accelerate our research.
So your team just recently developed a new biosensor for detecting Lyme disease. How would you explain to a 5-year-old what it does?
To a 5-year-old, yeah. So Lyme disease is caused by a bacterium. I'll call it a germ, just to make things simple. And normally, we don't know. We can't see, of course, when someone has that germ in them, which makes it really difficult. And so today, if someone was to get tested for Lyme disease, we would ask the body, have you seen this germ? And that's really not very reliable. So what we've done is we've basically asked whether we can see the germ itself by using tiny little electrical signals. So the same type of little zap of electricity that you might get when you walk across a carpet and touch something in the winter. When our sensor sees the piece of the germ, it'll create a little electrical signal, basically.
Very cool. And what initially inspired you to study Lyme disease?
So I have been interested in the life sciences since I was in high school. And I just continued to follow my curiosity into graduate school. I really just dove into my passions and really wanted to get into cell biology and microbiology. But that always intersected with my identity as someone who's lived with a chronic illness for most of my life. And when I was bouncing around between specialists trying to find answers and trying to heal myself, the question came up of whether I could have had Lyme disease, whether that was the instigator of a lot of my symptoms. And when I started to dig into that question, I, being a scientist, went into the literature and I started to dig around and try to really understand the problem from that perspective. And I realized all of the gaps that existed, the potential that was there to fill those gaps, both as a scientist and as someone who was coming at this from lived experience. And so I felt that what I had learned in my PhD was directly applicable to this. What I had learned, what life had taught me, was also directly applicable. So I thought it was a natural evolution.
And so what excites you most about your current research?
Everything. we have, I counted this, we have 14 different projects right now on the go. And we are tapping into expertise from microbiology, biochemistry, molecular biology, cell biology, entomology, epidemiology, and we're bringing all those people together under one roof. And it is so fun to have that kind of diversity and to have something that's truly interdisciplinary, to watch what comes out of people with somewhat different backgrounds working off each other, passing things back and forth, analyzing through different lenses. It's so much fun. It's so inspiring, and it gets stuff done. It solves a problem like this. And then you watch as projects start from pretty humble beginnings and they evolve. And sometimes those projects become their own programs. So you watch things as they kind of grow, and it's a lot of fun.
So imagine you have 60 seconds to convince a potential donor to invest $10 million, just a small sum of money, into your research. What's your pitch?
10 million sounds like a lot of money, but I want to put this in context because it was recently estimated that the projected cost to Canadian society of Lyme disease by 2050 is going to be in the billions. So over 2 billion was what Lyme disease will cost us. So I would say ounce of prevention is worth more than a pound of cure. Lyme disease isn't going anywhere until we do something about it. And we have seen people who have struggled with long-term complications. We have seen them become disenfranchised by the system and feel completely abandoned, essentially. And I also know that this is not something that's going to be a quick fix. There is no one magic bullet that's going to solve this. So we need a multidisciplinary program to look at this from multiple different angles. And that is something that we're developing, and that is something that we can offer, because this isn't a one cure type of problem. This is a complex problem that needs a complex solution and an entire program built around that.
Yeah. I'll sign a check.
Yeah.
What is one thing you wish people understood better about your research?
One thing that I think people generally find frustrating is how long it takes to go from our perspective, asking a question, formulating a hypothesis, collecting the data around that, analyzing the data, and getting it into the public domain, into a publication or a podcast or some sort of format that makes it palatable for people. It takes such a long time. And likewise, when we're dealing with something, as you mentioned, like a diagnostic approach, a biosensor, a device, the amount of time that we need essentially to develop that from a concept to putting data behind it on the bench and then taking that into a clinical space, testing it out with real patients, going through regulatory. It's a very long process, and it's not something that you can really cut corners. We know that people want solutions immediately. We want solutions immediately. It doesn't work on that timeline, unfortunately.
And so then on the flip side, what's one thing you wish you understood better about your own research?
Our own research, that's an interesting question. And I would say, I wish I understood better how this field of Lyme disease research ended up where it is, as a sort of neglected space. And also as a highly polarized space. And don't get me wrong, I do understand some of the drivers that got us where we are, some of the reasons that this field has fallen between the cracks. But here we are in 2026, and the question of Lyme disease and chronic illness is still very controversial. We don't have definitive mechanisms that people are rallying behind and universally supporting. We do not have treatments. And a lot of the issues that patients are bringing to us today are the same issues that caused patients to become advocates and activists in the 1970s, 80s, 90s, and here we still are. And it feels like we should be a lot farther along. And I'm not saying that we as taking ownership of this is a 40 or 50-year-old problem that unfortunately we have kind of inherited. But I wish that instead of silencing patient voices in the 80s and 90s and earlier, people had been more inspired to become more creative and actually solve problems.
And what do you hope to accomplish with your research in the next 5 years?
That's a great question. And we have so many things that are sitting there brewing, growing, evolving, that I have a long list of things to do in 5 years. But I think the big one that people want to hear about is probably the diagnostic strategies, because we've identified diagnostics as a linchpin issue in Lyme disease. And what I would really like to see happen is that a lot of these prototypes that we have developed in the lab that are doing really well at detecting Borrelia, the causative pathogen, in a controlled environment in the lab, those need to move into clinical testing. So actually using it on patients who are affected by Lyme disease, using it on people who don't have Lyme disease, and making sure that it's not accidentally diagnosing Lyme disease in that population. And then beginning to move it through the enormous task of regulatory testing, and eventually trying to bring something like that to market. So the next 5 years from that perspective is really getting these into clinical studies that will tell us whether they represent a viable path forward, and if so, what do we need to do to get those to the next step?
In your opinion, what do you think makes a successful grad student at your research lab?
I don't think there is one single model of success, and I don't want students to feel shoehorned into some sort of, perception of what a grad student should be. But I do think that there are traits and skills that are common among people who are successful in this environment. And I would say that starts with people who are just enthusiastic, keen, self-motivated. They want to ask questions. They want to learn. They want to contribute to this field. That goes a long way, especially when things don't go that well in the lab, which is a pretty common experience. You want that motivation to be intrinsic. I think creative thinking is really important, and moving away from this idea that you're consuming knowledge and being open and being capable of being in a place where you're now producing knowledge. I would also say emotional intelligence. And that's often not something that I think comes up a lot when you're talking about STEM, but the idea that people understand themselves, and that when they are working in groups, they can understand different perspectives. And they can also respect and have empathy for the people who are affected by the problem that we're studying. That goes a long way. And I think it's an underappreciated aspect.
So why do you think it's valuable to work with grad students?
So right now, we actually have a lab that's almost equally divided between senior research staff and grad students. And those of us who work in a mentorship capacity absolutely love working with grad students, because they bring an enthusiasm and an energy to this space, and an openness, I think, as well. They're just learning this. They're seeing things for the first time. And that's a lot of fun, to be around people who are just being exposed to new things. I think it also makes us better researchers if we're able to work effectively with them, to teach them, to help them navigate their path. And ultimately, we want to make great scientists. We want to contribute to people's journeys. to allow them to succeed wherever they go. So I don't have any expectation that people who come out of this lab will necessarily go into academia. So they have to be prepared to go into other fields like medicine. That's a big one. That's a lot of people who come through our lab do want to get into medicine and do get into medicine. Other professional schools, industry, and other kind of adjacent things. And grad school has to prepare them for that. And so if this lab for them is a formative environment, no matter what they go into, I think we've done our job.
Is there a colleague or faculty at the University of Guelph whose work you really admire?
There's a lot of them, but I would say Dr. Emma Allen Vercoe, who studies the gut microbiome. So when I was an undergrad at Guelph, she guest lectured in one of my microbiology classes. And I was so blown away and inspired by her work. And I loved this idea of transitioning from the kind of one pathogen, one disease model to the microbiome view of health and disease. And so I think I've always kept that in my mind since that lecture, this idea of complexity and community and essentially the sort of ecology of disease. I think that's been very foundational for me.
Well, if you could, so the next question, if you could only eat one campus meal for the rest of your University of Guelph career, what would it be?
I would be at the university club with that buffet every day. Yeah. Maybe that's a bit of a dodge of that question because it's not one single meal, but whatever they put out, it's so good. It's such a nice environment.
Okay, so last question, what does improved life mean to you?
For us, improved life isn't just a catchphrase. So when we go out, we do a lot of community engaged events, including fundraisers and visibility events for Lyme disease and for the foundation that funds us. And inevitably, during those events, we meet patients. They come up to us with their stories of Lyme disease, what happened to them, how it impacted them, how it was a complete game-changing event in their lives. And whatever we do, whether we're working on fundamental microbiology or technology or, clinical application, it is with people like that in mind. It is the idea that down the line, we are working towards a future where these problems are solved. So improved life to me means working towards a more hopeful, happy future.
Yeah, I like that. Well, thank you, Dr. Wills. I really appreciate you taking the time to chat with me. If you like what you heard, please like and subscribe to wherever you get your podcasts. For the latest updates, be sure to follow us on all our socials. And for more info, you can always visit uguelph.ca. We'll put all the links in the show notes, and we'll be back in your feed for another 15 with another fantastic faculty. Until next time, thank you so much for listening.